Differences between DNA and RNA is based on certain points like location, structure, types, function, etc. DNA and RNA are the types of nucleic acid. And we know that nucleic acid is the largest biomolecule. And if we compare the structure of DNA and RNA, the structure of DNA is very large. So that's why it is considered as the largest biomolecule. Location. DNA is present usually inside the nucleus. In the nucleus where? In the chromosome. And sometimes it is also found in the mitochondria and chloroplastid, which are the types of organelles, okay, which is present in the cell. Next is your RNA. RNA is usually found where? It is found in the cytoplasm, as well as it is found in the nucleolus, which is a part of nucleus, and sometimes in the chromosome. The next difference is regarding the structure. Okay, DNA is double-stranded. Double means two. And this type of double-stranded structure is called the helical structure. And RNA is single-stranded or linear structure. Over here, I have drawn a very simple figure of DNA and RNA. You can see that RNA consists of just a single strand. So this type of single-stranded structure is linear. Whereas in DNA, I have used two colors, okay, over here, green and blue. So this indicates that it consists of double-stranded structure which are overlapping each other. And this type of structure is called the helix or the helical structure. Now the next difference is also regarding the structure is that both DNA and RNA are made up of nucleotide. And you know that nucleotide consists of sugar, phosphate and nitrogen base. So there is some difference regarding the sugar also. So in case of DNA, the pentose sugar, that is 5 carbon atoms, okay, but the pentose sugar is deoxyribose. And in case of RNA, there is pentose sugar only, but the pentose sugar is ribose. I have already taught you the differences between ribose and deoxyribose sugar. The next difference is regarding the nitrogen base. The nitrogen base is of two types, purines and pyrimidines. The nitrogen bases of DNA is, in case of purine, it is adenine and guanine. In case of pyrimidine, it is cytosine and T for thymine. For RNA, the purine bases are adenine, guanine, and the pyrimidine bases are C for cytosine, U for uracil. Now, if we check both this, you can see that the nitrogen bases of DNA and RNA, the three bases are same, adenine, guanine, cytosine. Over here also adenine, guanine, cytosine. What is the only difference? The only difference is the highlighted one. In case of DNA, the nitrogen base, the pyrimidine part is thymine. But over here, thymine is absent. Instead of thymine, what is there? Uracil. So in case of RNA, instead of thymine, uracil is present. In case of DNA, instead of uracil, what is present? Thymine is present. So that is the main difference in the nitrogen base of DNA and RNA, which is very, very important.